my name is Sophia.
My name is Farhiyo.
I'm Isabella Mendez.
And I'm Anna Amaral, and today we'll be presenting invasive crab species in Maine, abundance, interactions, and harmful effects. To begin, let's get some general information and background information on what defines an invasive species. An invasive species is any species that is non-native or alien to the ecosystem it is introduced to. This means that the organisms within the ecosystem will not yet be accustomed to this new species. Invasive species can be introduced to new ecosystems from around the globe in a variety of matters. One example is they can be brought over as larvae in ocean-going ships. They can be released as bait, or they can be released as aquarium organisms. Invasive species have a lot of potential to harmfully impact ecosystems in many different ways. They can impact biodiversity and ecosystem services. When an invasive species comes to an ecosystem, there is a lack of native predators, which can lead to an overabundance of the species from over-reproduction, and they can overconsume a certain species within this new ecosystem. Because of this, this can disrupt biodiversity and ecosystem services.
Why Carcinus manus and Hemigraptus sanguinis? Carcinus manus and Hemigraptus sanguinis are both invasive crabs often found on the coast of Maine. Carcinus manus, also known as a green crab, shown on the top right-hand side of the screen, is one of the invasive species commonly found. It was first found in Maine in the early 1900s, but it's originally from the Baltic Sea, and it was first found there in the 1800s. It's currently affecting the population of Maya arenaria, but it's also quickly reproducing. The other invasive crab commonly found is the Asian shore crab, the Hemigraptus sanguinis, which was first found in New Jersey in 1988, but it is commonly found here in the intertidal zone in Maine. It's quickly reproducing, and it's also found in the intertidal zone, and you can see a picture of it on the bottom right-hand side of the screen.
Biodiversity is the variety of life in an ecosystem. Organisms rely on each other to thrive, and when there is an overabundance of one specific organism, it can set off the balance and harm the ecosystem. When invasive species such as Carcinus manus and Hemigraptus sanguinis invade an ecosystem, they have a negative effect on the biological diversity, which has a negative effect over the entire health of that ecosystem.
So what is the importance in addressing the potential impacts that invasive species has on the broader world? Well, not only are invasive species affecting our biodiversity and our ecosystem, but they're also taking a negative toll on our economy. They reduce productions of fisheries by affecting native species such as worms, clams, mussels, and other living organisms. This has significantly hurt the New England's softshell clam industry. In fact, according to the National Oceanic Atmospheric Administration, or NOAA, research conveying 2014 commercial catch of U.S. seafood on par with 2013 displayed that in the U.S. alone, over 50% of fisheries depend on the Atlantic coast for their economy. This means that if Hemigraptus sanguinis and Carcinus manus eventually increase in population, they can negatively take an impact on our fisheries, further giving a negative impact to our economy. So before we started our research project, we needed to get some background information about the Hemigraptus sanguinis and the Carcinus manus, so we came up with this research question. What are the interactions and differences between the Carcinus manus and the Hemigraptus sanguinis, and how do their abundance affect diversity within the intertidal ecosystem along the Maine coast? Not only did this question help us to read many science journal articles, it also helped us to conduct our hypothesis.
Using our research, we formed our hypothesis. If there is a high abundance of invasive species in the intertidal ecosystem, then it will be less diverse. Using our background knowledge of the potential impact invasive species can have on biodiversity, we believe if there was a large abundance of invasive species in one area, then it would be less diverse in correspondence. We went to 3 different locations along the coast of Maine: Recompense, Giant Stairs, and Rachel Carson Preserve. During our data collection week, the first day, April 4th, we went to Recompense in Freeport. The next day, April 5th, we went to Giant Stairs in Bailey Island. The last day we collected data, April 8th, we went to Rachel Carson Preserve. All three of these locations have rocky intertidals, which are the best locations to find invasive crabs. We chose these locations because they are separated, eat nicely, and we could have a broad view of the biological diversity along the coast of Maine.
In order to collect our data, we had to use Simpson's Diversity Index, which is an equation that we use in order to test the diversity in an area. We use the equation first by setting up the transects, and in order to use the equation correctly, we had to set up quadrants in order to test the diversity in those areas. Our group actually split up into pairs in order to test the diversity. While two of us were testing the diversity in a given area, like Sophia said, the locations in which we tested, the other half of us were collecting crabs. In order to collect the crabs, we looked for their sex, their measurement, also patterns that we saw, and often where they were in abundance. We then combined our information in order to find the abundance. We also used Simpson's Diversity Index, like I said before, which we used a scale where 0 was the least diverse and 1 was the most diverse, and we combined our information there in order to test the high abundance of the crabs that we often found on the coast of Maine.
As you see in the top right, we used a quadrat to find the general diversity of an ecosystem. A quadrat is basically a square where we can see the general biodiversity in one area, and then from there we use Simpson's diversity to get a general idea of the biodiversity. To set up these quadrats, we set a transect, which is a vertical line to the rack line, and we set the quadrats 3 meters apart from one another.
Here we have a graph of the abundance of Hemigrapha sanguinus, also known as the Asian shore crab, found at Giant Stairs. Giant Stairs was actually the location in which we found the highest abundance of the Asian shore crab. We actually found 88 crabs there, and that is the highest abundance that we actually found in our whole research project. With the 88 crabs that we found 48 female and 40 male, 56% were female and 44% were male. As the graph shows here, the y-axis measures the abundance and the x-axis measures the size of the Asian shore crab. 2 was the most common found size for the male, 2 centimeters, and 1.5 centimeters was the most common found for female. And actually, we never found Carcinus manus and the Asian shore crab both together, so that also helped us think whether the Asian shore crab was actually dominating over Carcinus manus.
In this graph, you can see the total abundance of Carcinus manus crabs found in Recompense and Rachel Carson Preserve. On the y-axis, you'll see the total abundance, and on the x-axis, you will see the size of the crabs. At Recompense, 7 people searched for crabs in a time span of an hour, and only 1 female Carcinus manus was found. At Rachel Carson Preserve, 3 people searched for crabs in a time span of an hour and 50 minutes, and 3 male Carcinus manus were found. We found fewer crabs here than we did at Giant Stairs. Not only were we testing the abundance of the ecosystem, but we were also testing diversity. And later we saw how these two corresponded with one another. Here are the 3 graphs we made for diversity. As you can see, Giant Stairs, we used 3 transects rather than 1 because the features of the ecosystem and the organisms were much more diverse as we moved down the ecosystem, while Recompense and Rachel Carson Preserve were much more consistent. Giant Stairs was our most diverse area, although we found the most high abundance of invasive species here, while Recompense was our least diverse ecosystem. In fact, in Recompense, we only found 1 quadrat that had any diversity at all, while the other 2 quadrats only contained dead organisms. We found it very interesting that Giant Stairs was the most diverse area while having the largest abundance of invasive species, because typically, like our hypothesis stated, we believe that the diversity would be lower as the higher abundance of invasive crabs were there.
This is also another graph we created after data collection week, and in this graph is a regression line. So basically this regression line is simply indicating the values and relationship between diversity and invasive crab abundance. This statistical measurement gave us 2.4% correlation between diversity and crab abundance. This simply means that there was no correlation. So, strongly implying that areas of higher biological diversity did not correlate with crab abundance. Overall, we can conclude that the highest abundance of invasive crabs were found in the ecosystem with the highest diversity.
Additionally, we found it concerning about the abundance of Asian shore crabs we found, and we found potential that they may be outcompeting Carcinus mayonnaise. Because they were in an area of the highest diversity and the largest abundance, a much larger abundance than that of Carcinus mayonnaise, and they have been here for a shorter time, we can be led to believe that perhaps the reason they were here is because invasive species are being more lured towards the areas of higher diversity, and this may have a major impact in the near future, and may also show that they are outcompeting Carcinus mayonnaise. There may have been other reasons why we found a larger abundance of Asian shore crabs at giant stairs, such as organisms present, but from our research we can make an inference that perhaps the reason why they were there is because they were more connected to the area of higher diversity, which could pose a major threat to Maine's coast in the future. We have so many people to thank for the success of this project, and we'd first like to thank Carrie Whitaker for the amazing support and guidance she gave us from running through the mud with us to find crabs, very carefully teaching us how to use Excel, and teaching us that all data is good data.
We would also like to thank Maddie Halloran for helping us with all our questions that we had, helping us look for crabs, and always being there when we had any questions to ask.
We'd also like to thank our another amazing RA, Cordelia Raw, for editing our papers and looking at things from a non-scientific sense of view.
We'd like to thank our humanities teacher, Rebecca Turkowitz, for editing our science papers and giving us support.
We would also like to thank Kate and Jenny for helping us understand the importance of feedback, and Gemma and Razel for helping us collect crabs in the muddy field. Questions? Yeah.
You had said in one of the areas where there was very low diversity, you had found a lot of dead critters.
Yes.
Was there anything to indicate that this sort of morbid site had an invasion of one of the invasive species? What were the kinds of organisms that you found that were dead?
Primarily, we found shells of Mytilus edulis, and we found some empty Luterina shells as well. We found, the blue mussel and the common periwinkle. And we believe that perhaps the reason why there was a lot of dead organisms there is because the green crab has been established in the Maine coast for a long time, and they may have already had a major impact on diversity. But we do believe because of the abundance of Asian shore crabs coming in, that perhaps Asian shore crabs could even make a bigger difference on the ecosystems along the Maine coast. Thank you for your question.
Suppose I hired you today to go one step further. What would your next step be in this project? It's kind of a out there question, but.
If we had to do a next step, so we have the data that shows invasive crabs and diversity, and so our questions are now, which crab is more abundant on the coast? In our data, it was the Hemigraspis sanguinis, but what if we stretched out farther in our data and looked at different areas of the coast of Maine and really try to compare all the locations, then maybe we could see a change. And also, like Isabella pointed out earlier, in most of the regions where we found Hemigraspis sanguinis, there wasn't any Carcinus mayonnaise, so there's this dominant kind of effect that Hemigraspis sanguinis has on Carcinus mayonnaise that we've observed, and we would like to see that more.
If there aren't any further questions, let's give them a round of applause.